Okay, we're going to talk about meiosis 2 now. All right, so let's just step back for a second and look at where we ended up last time. So we were talking about meiosis 1, the first round of division in meiosis, and we started out with 46 chromosomes, actually 46 chromatids until it doubled into 46 chromosomes. So we're just going to start there. Remember, again, we are talking about germ cells. The output of meiosis, the whole thing, the purpose is to make gametes. Okay? So we've got germ cells in the reproductive organs. We are in the reproductive organs of an organism, and we find a germ cell. It's got 46 chromosomes, which makes it a diploid, or 2N. So throughout meiosis 1, we've got our first round of cell division, and we come out with 23 chromosomes. All right, and in there, we also have the unique event of synapsis. Remember, that's where crossover happens. So now we've got two cells that are haploid because they're half of what the parent cell had. Now, this is a tricky part going into meiosis 2. Meiosis 2 is really easy, except this can be kind of tricky. So I'm going to clear this up right off the bat. The end product here, so what we're going to do in meiosis 2, so this is where meiosis 2 happens, is each of these cells divide. And your text, many texts, a lot of texts, are going to say that the product is 23 chromosomes in each of these, and that they're all haploids. They're N, so they're haploid. Now, if you look at this, how in the world can we have 23 chromosomes here, 23 chromosomes there? It's haploid, and I'm going to tell you this, in interphase 2, there is no division. The DNA doesn't duplicate anymore. It's done. So how do we get 23 and 23 still have haploids if 23 haploid divided? That can be tricky. That's one for the ages, huh? Wrap your mind around that one. Well, what happens is that this is actually, it starts out as chromosomes, and what they should be telling you is that we've got 23 chromatids in each of those. That's how it's a haploid, because it's still half of what the parent had. We still have 23 chromatids instead of 23 chromosomes. So it's a haploid because it's half of the parent cell, and it's 23 because we're talking about chromatids instead of chromosomes. So I don't want that to be confusing when you're reading, because the texts will often refer to this as 23 chromosomes, 23 chromosomes, it's a haploid, and they don't bother to tell you why you can have 23 here and 23 here. It's because this is a chromosome, that's a chromosome, that's a chromatid. Big difference. Okay, so now that's done, let's talk about some meiosis 2. So just like we said, we ended with two cells that had 23 chromosomes each. All right. During interphase 2, most cells don't go into interphase 2. The ones that do not replicate anymore. So then we go into prophase. So we've got a cell with 23 chromosomes. Now it's going to be just like mitosis. So in prophase 1, we still have the centrioles appear, the chromosomes, or the DNA condenses to form chromosomes, and the spindle fibers appear. It's all the same as mitosis. Then in metaphase 1, now our chromosomes line up in the middle, just like in mitosis. The only difference is that now we're talking about germ cells. So they line up in the middle. Then we go into anaphase 2. And there's a little difference here. There's something that you're definitely going to have to understand. I think I've been writing one, haven't I? 2, whoops. 2 and 2, sorry. So anaphase 2. This time, so in anaphase 1, just a side note, it's good to compare the two. Anaphase 1, we had the homologous chromosomes being pulled apart. This time, this is like mitosis, where we have the sister chromatids being pulled apart. So we're going to have this again. So anaphase 2, sister chromatids are pulled apart compared to anaphase 1, where it was the homologous chromosomes. Remember, we had one from mom and one from dad being pulled apart. So that was anaphase 1. This is anaphase 1. This is anaphase 2, just like mitosis. All right, then we go into telophase. Again, the super easy thing about that, if I can get this to cooperate, is that it's just like telophase and mitosis. So we have telophase 2. In telophase 2, now we've got our chromatids at either end of the cell, and it forms 2 nuclear membranes, and then cytokinesis is dividing the entire cell into 2. So then we see with cytokinesis it pinching in, and we end up with 2 cells. So let's just go back through everything with a nice comparison. So we've got meiosis 1. We're going to start out with 46 chromosomes. And once it doubles in interphase 2, it's a full chromosome. And these are germ cells in the reproductive organs. It's 2N, which makes it a diploid. All right? So in meiosis 1, we have this division. A very special event, cannot forget this, synapsis, is in prophase 1 of meiosis 1. A very important event where we have crossover for genetic diversity. Now what comes out of meiosis 1 are 2 cells with 23 chromosomes each. Now they are a haploid because they're half of the parent cell. Now we go into meiosis 2. Where these 2 cells now go through all of the stages of prophase, metaphase, anaphase, telophase. And we come out with 4 cells now. 4 cells that now have one chromatid from each of the chromosomes. So it's 23 chromatids instead of 23 chromosomes. Even though the text might say 23 chromosomes, remember, when we finish meiosis, those are chromatids. And they're still called haploids because it's half of the parent cell. We still have half the chromosomes of the parent. So what we end up with are 4 daughter cells that are unalike. So they're all unique because of crossover, because of reorganization. They're not alike. Now let's compare that to mitosis. Mitosis, we start out with 46 chromosomes. And here in meiosis, we were talking about sex cells. In mitosis, it's all other cells, anything else. And we start out 2N. It doubles its DNA, it divides, and we end up with 2 cells that are identical with 46 chromosomes each, and we still call them daughter cells. We can put 2N because it has the same as the parent. So there's the difference between meiosis and mitosis and kind of the whole process of meiosis wrapped up. Good luck on your test.